Okay, then we are all silent. Also the grass mower. No, he doesn't listen. Welcome to Easy Immunology. My name is Stefan, and in our today's lecture we want to talk about T cells. And in order for me to not do this completely alone, I have today again with me Alex. Hi Alex.
Hi Stefan.
Hi, thank you very much for joining me. And I have with me Hannah. Hi Hannah.
Hi Stefan.
Thanks again also for joining me. The two will support me with questions, so they can ask the questions that you might have also during this recording, and then we can address them directly here in this video. And this, as we learned, is very helpful for everyone. So as I said, we're going to talk about T cells. We are starting with a teeny tiny bit of repetition at the beginning. So what we learned in the first video is that our immune system protects us, usually very efficiently, from pathogens. So these are microorganisms or viruses that want to make us sick because they want to grow inside or on our body, on our costs. And our immune system, as I said, protects us from these pathogens. And it, broadly speaking, consists of 4 different components. We have learned the last time that there are the anatomical barriers that just protect us from these pathogens by preventing the pathogens from coming from the non-sterile outside into the sterile inside of our body. If the pathogens manage to come inside, they are attacked by the complement system and the antimicrobial peptides. And if this also doesn't work, or in parallel to this, they are also attacked by cells of the innate immune system. This is what we managed to go through in the first video, and today we are starting at looking at the adaptive immune system, so which are the T cells and the B cells and the antibodies produced by B cells. And this is very, very complicated, we have to say. Therefore, we were able to look at the first 3 barriers in the first video, and for the last barrier, the adaptive immunity, we will probably need 3 videos to go through all the concepts. But it's very important because the first 3 barriers are able to eliminate 95 to 98% of all pathogens, but the last 2 to 5% for which we need the adaptive immune system, for this we actually need it, because there are patients who do not have an adaptive immune system and they are not able to live in a normal environment with all these pathogens, because the adaptive immune system is so crucial in eliminating them. So what we also learned, I think this is where we stopped the last time, is that the innate immune system, which consists of different cells that we looked at, is available immediately after birth. It can react within minutes and hours, and it does so by recognizing the pathogens as dangerous and foreign using a limited number of pattern recognition receptors. We looked at the different classes, and this is rather effective. As I said, eliminating 95 to 98% of all pathogens, but broadly speaking, it doesn't remember what it did yesterday, and therefore if you are reinfected with the same pathogen, you have to start the whole cascade again, and this is not the most efficient way to do this. And what we want to do over the next 3 videos is look at the adaptive side, and we are going to start with looking at the cells. As we learned in the last video, the innate immune system consists of different cells. We have looked at the dendritic cells and macrophages as sensor cells that recognize pathogens. We have looked at the eosinophils, mast cells and basophils, which are able to fight big extracellular pathogens like parasites, multicellular parasites that can be very, very big, up to 12 meters in length. And we looked at the neutrophils, which are very important, the most frequent cells in the human blood, because they are able to fight pathogens with many different effector mechanisms. We didn't look at the NK cells and the NKT cells yet. We will do this at the end of the following video, because we need to first understand the T cells, and then it's very easy to understand the NK cells and the NKT cells, because if I spoil you, these are like evolutionary precursor cells to the T cells we are going to start looking at today. On the adaptive side, we have the T cells. There are different types we will look at today, and together they are referred to as the cellular part of the adaptive immune system. And we have the B cells and the antibodies produced by B cells, and they are referred to as the humoral part of the immune system. The terms cellular and humoral are historically grown. They are not totally correct because, as you can see, the cells of the adaptive immune system are not concentrated in the cellular part. So the cellular part is just the T cells, but also the B cells are cells in the end. So they are also cellular. But these are historical concepts because, as we will see, it is very hard to differentiate T and B cells with just a light microscope. So there's basically no difference unless they are activated. Therefore, people couldn't keep them apart from each other. And what people were able to do is they were able to draw blood and analyze the blood, and they found like the antibodies in there. And therefore they said, okay, the antibodies are in the humerus. This is like the bodily fluids, and they protect us from many pathogens. So the antibody must have something to do with the humoral fluids, and therefore it was called the humoral immune system. Okay. So today we are going to focus on T cells, and there are 3 different types of T cells we are going to look at in more detail in the next slides. So first, here is like a lymphocyte. So T and B cells together are called lymphocytes among a lot of red blood cells. The red blood cells are the pale ones in this picture. And you can see that they are like disc-shaped, and they do not have a nucleus. They do not have a core inside the cell. And you have the single big lymphocyte, which consists of the nucleus. That's the pink part. And the more pale part around is the cytoplasm. And you can see that you basically cannot see a lot. It is a round cell without any characteristics. And it has a very big nucleus and only very little cytoplasm. So that's what you can see, and that's why people were also not able to distinguish between T and B cells, because at this stage a T cell just looks like a B cell, and it's very hard to distinguish if you do not have, like, for example, genetical markers that you can look at. On this level, of course, you can distinguish a T cell from a B cell. They are very different. In terms of stages, we can differentiate 3.5 stages for T and B cells. We have the precursor cell, that is the gray cell, that is like the lymphoid progenitor cell that develops into either a B cell, that is the left part, the blue part, or a T cell, that is the green part. And we can differentiate between naive T cells and B cells. These are the small round guys that I showed you. And they are, as I said, very hard to distinguish because there is not much to see. Once T and B cells get activated, they differentiate, so they change. You can see actually they look very different. They are much bigger, and they proliferate. So they form a lot of identical daughter cells because these cells are needed during the infection to fight the respective pathogen. So these are the activated T and B cells, and there you can already clearly see differences with a good microscope between a T and a B cell. And then once they find the pathogen and start to fight it, they fulfill their so-called effector function. For the B cell, it is producing the antibodies. You can see the B cell is surrounded by a lot of antibodies in our case. And for the T cell, it is, at least for this type of T cell, it is the production of cytokines, so messenger molecules that direct the immune response. There you can also find differences. So these are the 3.5 stages for the T and the B cells that we can differentiate. So where do the cells of the immune system come from? Where do they develop? They develop in the so-called primary lymphoid organs. This is the bone marrow and the thymus. And in the bone marrow, we have so-called pluripotent stem cells. So these are stem cells, are cells that can develop into many different cell types. They are not able to develop into every type of cell that would be omnipotent, like the fertilized oocyte is able to and has to develop into all cells in our body. This is less potent, if you want to call it like this, and it develops into first the common myeloid progenitor, and this then forms or is the predecessor for most of the cells in the innate immune system, like dendritic cells, macrophages, neutrophils, and the granulocytes. And the common lymphoid progenitor develops in the bone marrow into the B cell progenitor and then into the B cell. and it can migrate, the common lymphoid progenitor can migrate to the thymus and there become the T cell progenitor and develop into the T cells. And Alex has a question.
Is the B cell called B cell because it comes out of the bone marrow? Like the T cell comes out of the thymus?
Yeah, one could assume that, and it's a nice way to remember it, but actually not. The B cells are, for some crazy reason, called B cells because they develop in the bursa fabricii. This is an organ in chicks where they were first developed because some guys did, or some guys and girls did experiments on chicks. So this is not why they are called B cells. Okay?
But you can remember them quite good.
It's
with bone easy. Marrow is B cell and thymus is T cell.
Yeah, but in the end, if we are 100% correct, it's not correct. Yeah. Okay, so today we want to look at the T cells. And this is an overview how actually an adaptive immune response is formed. And it starts with a pathogen being recognized by an antigen-presenting cell. We looked at this in the last lecture already. And then it activates, this activated antigen-presenting cell activates a naive T cell that turns into a effector T cell. And this then activates B cells and they turn into antibody-producing plasma cells. And we're going to start with the first step, the activation of the naive T cell. And here we have our antigen-presenting cell. Usually this is some kind of dendritic cell or also can be a macrophage, but usually it's the dendritic cells. They have so-called pattern recognition receptors on their surface or in their inside also. And these receptors are able to recognize certain structures. They are called PAMPs, pathogen-associated molecular patterns. And these are structures that only exist in this shape or form in these microorganisms. And in contrast, they do not exist on our own cells and tissues. So this is something that is definitely foreign. So if these receptors bind, these pattern recognition receptors bind to these pumps, this means that something foreign is around. And this, for this cell, means just, okay, in this case, here there's a bacterium around which has a flagella, and therefore an immune response needs to be made against this bacterium. So here we have the flagella protein, it's called flagellin. It binds to the pattern recognition receptor, and this triggers different things in our antigen-presenting cell. First is a change in gene transcription. So there is, through a very complex set of adapter molecules, a activating signal transduced into the nucleus, into the core of the cell. And this results in many, many different genes being turned on or turned off. So if you have a antigen-presenting cell that is infected by a virus, this you can do in experimental settings, thousands of genes are up or down regulated. And if you remember that we only have between 19,500 and 23,000 different genes, thousands, 5, 6, 7,000 genes that are up or down regulated is quite a lot. Among these genes that are upregulated are genes that code for so-called co-stimulatory molecules. These are molecules that are then expressed on the surface of the antigen-presenting cell, and they are only expressed there if the cell has recognized something dangerous, something foreign and dangerous via its pattern recognition receptors, and therefore the presence of these molecules is just a way for the antigen-presenting cell to show to the T cell that it wants to activate that it has found something dangerous. For the T cell, it doesn't matter what the cell has found, it just needs to know, okay, there's something dangerous around. Also, among the genes that are activated are genes that encode for the production of cytokines, small soluble messenger molecules that transmit information between immune cells and non-immune cells. Additionally, the cell either triggered by the activation of the pattern recognition receptors or also generally takes up everything that floats around the cell. So these cells are like PhD students, they are always eating, and these cells just eat everything around them, and including whole pathogens or parts of the pathogen. And these molecules are then inside the cell digested, turned into peptides. So proteins are long polypeptides, and they are cut into short fragments, and these fragments are called peptides, and these peptides are presented on so-called MHC molecules, major histocompatibility complex proteins. We will look at them in the second half of the lecture once we have understood what a T cell is and what kinds of T cells there are. And then the antigen-presenting cell, shows on its surface what it has taken up. That's why it's also called an antigen-presenting cell, because it presents peptides derived from the pathogen molecules, usually proteins, on its surface. And then the cell that is activated moves from the place where it's found the pathogen. This can be everywhere in our body. It moves to a lymphoid organ. This is usually a lymph node where the T cells are also wandering around. So the T cells are wandering around in our body. It takes 24 hours approximately for a T cell to reach every lymph node in our body, and the antigen presenting cell goes there and just waits for the T cell. So there are many T cells coming and going, and every T cell checks what the antigen presenting cell has to offer. So it has like this T cell receptor. I'm gonna highlight it in post. This is the gray and green structure, and this is able to recognize the peptides from our blue bacterium together with the MHC molecules. And if this matches, this means this is the T cell that was born, if you want to say it like this, to fight this blue bacterium. And this just results in the two cells sticking together. It's not enough, and it's not strong enough to stick together long enough for the T cell to become activated. And this is where the costimulatory molecules come into play, because they result in the two cells sticking together even more strongly and even longer. And both signals then transmit an activating signal into the nucleus of the T cell, and then it can be activated. So it knows via its T cell receptor that the T cell receptor it has is useful for fighting this blue pathogen. And via the costimulatory molecules, it gets the information that it is allowed to fight the pathogen, because the antigen presenting cell has decided that the blue bacterium is dangerous and foreign. But it doesn't know what to do, and the information what to do and what kind of T cell to become is coded in the cytokines. So these cytokines bind to receptors on the T cell surface, and then they trigger the differentiation, so the professionalization, if you want to call it like this, of the T cell into different types of T cell, because it could become, for example, an antibacterial T cell, an antiviral T cell, an antiparasitic T cell, an antifungal T cell. This is basically what the immunologist calls the three-signal theory of T cell activation. And the first signal is the presentation of peptides in combination with MHC molecules. The second signal is the expression of costimulatory molecules, and the third signal is the production of cytokines. And as I said, the signal one is the binding of the T cell receptor to the MHC complex with the peptide. The signal 2 is the expression of costimulatory molecules, and the signal 3 is the secretion of cytokines. And what these signals do is, signal 1 ensures that only a T cell specific for a certain pathogen gets activated. Signal 2 makes sure that this only happens once the antigen presenting cell has decided that this cell is dangerous, because only then we have the expression of the costimulatory molecules. And signal 3 makes sure that the T cell is aware and knows what to do and what kind of T cell to become. So unfortunately, as I tell my students all the time, it is more complicated because there are different types of T cells that can develop. So we have 3 types of T cells. We have helper T cells, we have regulatory T cells, and we have cytotoxic T cells. And I want to just go through them one by one and explain you what they do and what their distinct functions in the immune system are. So let's start with the helper T cells. They are CD4 positive. This is something that we will look at in the later half of the lecture. But once they are activated by the the three signals that we just discussed, they have their main function in producing cytokines. They produce more cytokines and different cytokines than the antigen presenting cell did after activation, and these cytokines are then directing, modulating, and maintaining the immune response. And they can activate, for example, macrophages. These are cells that are able to phagocytose, to eat up the pathogens and destroy them and induce inflammation. Also, neutrophils, as we learned in the first lecture, are very important cells that induce inflammation. And they can also activate the other kinds of T cells, the cytotoxic T cells. These are the red guys. And they can activate B cells. In a way, these CD4 positive T cells are the managers of the immune system. So they tell all the other cells, or most of the other cells, what to do. And then they can coordinate in this way the immune response. Unfortunately, it gets even more complicated because there is not just one CD4 positive T helper cell. There are different types, and it is even more complicated than I have put it here on this slide. On this slide, I've put 5 different types, but there are even more, as Alex and Hannah know. What kind of T helper cell is generated depends on the signals that are transmitted by the antigen presenting cell during the activation of the naive T cell. And if, for example, the antigen presenting cell produces the cytokines interferon gamma and IL-12, this results in the binding of these molecules to their receptors on the T cell surface, and this triggers the activation of molecules inside the T cell. They are called STATs, signal transducers and activators of transcription, and there are many different STATs. And interferon gamma and IL-12 lead to the activation of STAT1 and STAT4, and this triggers the activation of a transcription factor that we call TBET, and this is called the master transcription factor of Th1 cells. So if cells activate this TBET, this then in turn activates many different genes in this T cell, and this T cell then turns into a so-called Th1 cell, which in turn produces a lot of interferon gamma. And this interferon gamma tells the immune system that there are intracellular bacteria that need to be fought, and this is how they then fight these bacteria, by activating all these other cells. In contrast, if the antigen-presenting cell or some other cell provides interleukin-4, this triggers the activation of STAT6, which in turn activates the master transcription factor of Th2 cells, which is called GATA3. These Th2 cells then produce IL-4, IL-5, and IL-13, and their main function is to coordinate the elimination of big multicellular parasites. And in turn, we have, for example, Th17 cells. They depend on raw gamma T. They are responsible for coordinating immune responses against extracellular bacteria. We have follicular T helper cell. They are mainly in the lymph nodes, and they help B cells to become activated. They provide the perfect environment for the cells in the lymph node to become activated, to proliferate, and to start immune responses. And we have regulatory T cells, which is like a special kind we will look at also in more detail later, because their function is not to induce immune responses, but to shut them down. Because once you have started an immune response, have eliminated pathogen, you also have to stop making the immune response at some point, because this takes a lot of energy, this takes a lot of resources, and if the pathogen is no longer around because it has been eliminated, you don't need this anymore. And this is what the regulatory T cells are for. We have also the cytotoxic T cells. They are CD8 positive. They are induced if the antigen-presenting cell produces interleukin-7. This triggers STAT1 and STAT4. You see, this is very similar to the Th1 cell. They are also like a Th1-like phenotype, because they probably were the same cell during evolution, and then this diverged into these two different cell types. They can produce also a lot of interferon gamma, but also perforin and granzyme, which they use to kill intracellular pathogens, virus-infected cells, and tumor cells. But we will look at this in more detail in one of the next slides. So these are the T helper cell. Their main function is to produce cytokines, and these cytokines then coordinate the immune response in eliminating the pathogen. Then we go to the CD8 positive cytotoxic T cells. Their main function is to kill virus-infected cells. So here we have a normal cell in our body, for example, a liver cell that is happy, producing liver enzymes. And also these cells are able to show on their outside what they are working on in their inside. They also have these major histocompatibility complex proteins. You can see they look a bit different than the ones on the antigen presenting cells. Why this is the case, we will look in a few slides. But also these cells also always show on their surface what they are working on the inside. And if they just are happy and work on liver enzymes, everything is fine, then the T cell will not do anything. And what the T cells are doing, they are circulating in our body and check what kind of peptides are presented on these MHC molecules. And if this is just a liver peptide from a liver enzyme, then everything is fine because the cell is doing what it's supposed to do. Yes, Hannah?
Does every T cell only have one T cell receptor for one specific peptide, or can they recognize more peptides?
Yeah, that's an excellent question. We would have gotten to this at a later stage, but we can also just quickly spoil it here. You can see on the surface of the T cells, there are 1, 2, 3, 4, 5, 6, 7 T cell receptors. They are all identical. So one T cell only expresses one type of T cell receptor, but we have millions of different T cells, and everybody has a different T cell receptor because this is generated randomly, as we will learn in the second half of the lecture. And it can only recognize, I wouldn't say one specific peptide, because peptides are in the end are just a certain amount of amino acids next to each other, and there are different amino acids that have similar properties. So it can be that one T cell receptor can maybe recognize 2, 3, 4 different peptides, which are not totally different, but they are differing only at certain positions in their sequence, and there they only have very similar amino acids which have very similar biochemical properties. So, but in theory, a T cell receptor is very limited in what it can recognize. Only a very small subset of very similar peptides. Okay. So if our cell gets now infected by a virus, the virus forces the cell to produce more virus proteins. And then we will have not only the pink liver peptides on the liver cell, but we also have blue peptides generated from the blue virus proteins. And now the T cell says, no, this is not correct. This doesn't belong here. You are very likely infected and I will kill you. And it does so by producing, among many other molecules, 2 molecules which are called perforin and granzyme. And the perforin just perforates the cell. This is, as you can imagine, not very healthy for the cell. And what is also not very healthy is the granzyme, because it induces apoptosis. So this is directed cell death. And this results in basically the virus-infected cell getting a signal to kill itself. And it does so because it is trained to listen to the signal of the T cell, and then this cell kills itself in a very controlled way without releasing all of its content and all the viruses inside the cell, just in the surrounding. And this is a very elegant way to very specifically and very targeted killing of just this one virus-infected cell in the middle, and the one on top and the one on bottom are not affected by this. This is just the function of the CD8 positive T cells, and that's why they're also called cytotoxic. Toxic means something dies or something gets damaged, and what gets damaged is the cyto, so the cells are killed by these cytotoxic T cells. That's why they're also called killer cells. Here I looked a long time in the internet to find a nice copyright-free picture. Here we have a cancer cell in the middle, and we have 3 T cells around it. And what was stained here is in blue is the nucleus, so the genetic material which is in the nucleus, and in green we have the cytoskeleton. So this is just it's like kind of like the bones or the muscle of the cell, the proteins they use to keep their shape and to migrate around. And in red, cytotoxic granules were stained. So these are small vesicles, small bags filled with toxic molecules. And you can see that there are no toxic molecules in the cancer cell, but they are in the T cell. And the T cells surround the cancer cell and they concentrate these toxic granules at the contact area to the target cell, to the cancer cell. And then they release the content of these cytotoxic granules, and this will lead to the death of the cell. So this is what we call an immunological synapse, because there is a contact formed between the cytotoxic T cell and the target cell, and this results in a very specific and very targeted killing of this cancer cell in this case. So the cytotoxic T cells kill infected cells, and then we have the regulatory cells to look at. And here we have again an antigen-presenting cell that wants to activate a T cell. You can see there is a peptide presented and a co-stimulatory molecule presented, and there are also some cytokines, which I was too lazy to put into the slide. And this cell would get activated because it gets all three signals. And if we imagine that this is, for example, an autoreactive T cell that would then start to run around and mindlessly kill our own cells in our body, then we do not want this. And this is where also the regulatory T cells come into play. They recognize the same peptide. It's again like the pink liver peptide from the liver cells. And they recognize it a bit differently. We will look at this also later. And they also bind to the antigen-presenting cell, showing the peptides on the MHC molecules, but now it transmits a deactivating, a suppressing signal to the nucleus of the antigen-presenting cell. And these molecules that they use to transmit the suppressing signal are co-inhibitory molecules. And they are somewhat the contrary molecule to the co-stimulatory molecules. We already learned about the co-stimulatory molecules. These are the orange guys that the antigen-presenting cell uses to activate the T cell. And here I put some example like CD40, CD80, CD86, where CD means cluster of differentiation. This is just a way of delineating that this is a molecule expressed on an immune cell, also a non-immune cell, and they bind to certain counterparts, certain receptors, and they are called, for example, CD40 ligand in case of CD40 or CD28 in the case of CD80 and CD86. Co-stimulatory molecules result in the activation of the cell, and then it can fulfill its function, but there are also co-inhibitory molecules. This is like the purple one, and they are called, for example, CTLA4 or PD1. PD1 is, for example, short for programmed death 1. And they bind to ligands like CD80 and CD86 and PDL1 or PDL2. And you can see that there's a certain overlap. I directly addressed this before one of the two ladies asked me the question. CD80 and CD86 can either be co-stimulatory molecules, or they can also be co-inhibitory molecules. It depends what they bind to and how strong they bind to it. So it's a question of avidity, how strong it binds. If it binds very strong to its counterpart, this is usually a co-inhibitory signal. But if it's binding not so strong, or if the counterpart like the CTLA4 is not present on the other cell, it is a co-stimulatory signal. And this results then in T cell inhibition. So this T cell will not be activated and start killing our own cells. In addition, the regulatory T cell, which is a subtype of the CD4 positive helper cells, as we learned, can also produce cytokines, so small soluble messenger molecules, and they are also suppressing the T cell activation. And do this into two ways. First, we have suppressive cytokines, and examples are interleukin 10 or transforming growth factor beta, TGF-beta. They just act on receptors on the T cells, and they suppress here this activation of the T cell. So if they get triggered with these suppressive cytokines, they just cannot be activated. So this also happens not only from the regulatory T cell to the effector T cell, this also happens from the regulatory T cell to the antigen presenting cell. This basically shuts down the antigen presenting cell because it does something stupid here. It tries to initiate, for example, an autoimmune response. Then we have another set of soluble molecules. So the regulatory T cells can also produce perforin and granzyme, and as we just learned, this will just lead in the T cell that it gets confronted with these molecules to undergo apoptosis. So then the cell is not there, and this is the difference. In the upper part, the cell is still there, but it cannot be activated. This is what we call energy. So this is a non-reactivity, even if they become stimulated by the next antigen presenting cell in the next lymph node. Whereas the lower part is called deletion. There the autoreactive T cell is not there anymore, and this is just a important distinction. And Hannah has a question. Yes.
Yes, my question is energy reversible, or
it is? Sorry? Yeah, it is reversible. So if, for example, a certain amount of time elapses or the cell gets very strong signals to become activated, it can be reactivated. But I know this is going to be the next question. You probably ask me, why is this the case? Nobody knows at the moment. Because one would say, okay, if the cell is autoreactive, the best way is just to shoot it in the head and get rid of it. Why do we keep the cell around that has no function? It is the case. We notice this can be very elegantly shown in animal models. In humans, it's very difficult to show this because it's difficult, let's say, for ethical reasons and technical reasons. But we know this is the case, and we do not understand it at the moment. Okay.
Do you think this is due to epigenetic modifications that we turn the energy?
Yeah, I think so. Yeah, I think so. So epigenetic modification means that the genes are modified. So there are ways. It's not a genetics lecture here. We are also not geneticists. Sorry, I don't want to lie. I am a geneticist, but I've stopped working in genetics like 20 years ago. There are ways to like constantly over long periods of time modify the way how the genes are used in these cells. That's what epigenetic means. Everybody has the same set of genes, but we can modify it how they are used, how they are activated, how they are not activated. And this is probably something that happens because these cytokines that induce energy, they bind to receptors, they trigger a lot of signaling in the cell, and this has of course influences on the genes in the cell. And this is how this is probably manifested over long periods of time, because we will learn this also later on. These T cells, once they turn into memory T cells, they can live for decades. So this is something that needs to be maintained for very long periods of time. Okay.
Thank you.
You're welcome. So let's go on. So we have completed our trio of T cells. So we have the regulatory T cells that are able to suppress, to shut down both helper cells and cytotoxic T cells in order to, I always tell my students, also the best immune response has to come to an end at some point, ideally once we have fought the pathogen. So now I want to turn to the question how antigens are presented to lymphocyte receptors. So how do the T cells, and for the B cells it's very similar, recognize the antigens that they need to react to? And to understand this, I have to say it's very complicated, as always. We are doing this in a simplified way. But in order to understand this, you have to understand that there are two different types of pathogens. So we have intracellular pathogens. These are pathogens that have chosen to infect cells in our body, and then they try to multiply, to grow, to replicate inside the cells of our body. And this has certain advantages because they can hide, or at least try to hide from the cells of our immune system. And they're not so easy, for example, to eliminate via antibodies. And then there are extracellular pathogens. So these are pathogens that do not infect cells. They live outside of our cells, for example, in our blood, in our stomach, in our gut, which is outside of our body, of course, but for example, in our tissues, in our brain, but not inside the cells. And this is, as you can imagine, these are two different problems the immune system has to face and has to solve. And it is done, or it is coordinated by the antigen-presenting cell that we already know. And we will start with the left side first. So we have intracellular pathogens, and the antigen-presenting cells swim around everywhere in our body, and they just let themselves be infected, for example, by this virus. So they're just passively infected by just wandering around in an infected tissue. And then this virus triggers the activation of cells, of the antigen-presenting cell, by recognizing the pathogen via their pattern recognition receptors. This triggers again the production of many different proteins, including cytokines, co-stimulatory molecules, and parts, peptides from the virus proteins are presented on the surface of the antigen-presenting cell in a complex with so-called MHC molecules, major histocompatibility complex proteins of type number 1. And then we have all three signals to activate a T cell. We have signal 1, which is the peptide with the MHC. We have signal 2, which is the co-stimulatory molecules, and signal 3, which are the cytokines. And this now results in the activation of the CD8 positive cytotoxic T cells, and they then start to search for normal body cells, if this is again our liver cell on top, that shows virus peptides, the blue ones, as part of their MHC molecules. And if it finds one cell, for example, the lower left one, it then kills, or it induces the cell to kill itself via apoptosis before it can produce more virus proteins. And you can see this is very efficient because only the one cell here that shows the blue virus peptides gets killed, but the one on the right side that shows normal liver peptides does not get killed. Okay, so there are these MHC class 1 molecules that are used to present peptides that have been generated inside our cells, and they contain a alpha chain. This is the green one, and a beta-2 microglobulin. This is a small globular, so round protein that stabilizes the fold of the alpha chain, and the alpha chain forms a peptide binding groove where the peptide is sitting. So how do the peptides get on top of the MHC class 1 molecules? So here we have a antigen-presenting cell, and what this cell does, it's the same for all cells in our body. They make new proteins all the time, and once the proteins are old, they must be replaced by new ones. And also, protein production is a very complicated process. As you might know, a lot of proteins do not correctly fold into their very, very complicated structure. Around 30% of all proteins are directly defective from the start because they do not find the right folding. And these old, damaged, used, or misfolded proteins have to be degraded. And this is done by a system that is called ubiquitinylation. So there is a ubiquitin tag added to these proteins that should be degraded. This is done by a structure called the proteasome. This is like a big barrel where the proteins get defolded on one side, and then the peptide chain is fed through this barrel, and inside the barrel are active sites of proteases. It's like a scissor, and they cut this long polypeptide chain into small peptides. So it's like a wood chipper where you put pieces of wood on one side in, and smaller pieces of wood come out on the other side. And these peptides are then transported into the endoplasmatic reticulum by a molecule called TAP, transporter associated with antigen processing. And there they find empty MHC class 1 molecules. They bind to them, and then they are transported by the Golgi apparatus via exocytotic vesicles to the surface of the cell, where they bind to the T cell receptor of the CD8 positive T cell. And this not only happens for misfolded proteins, but also for virus proteins that are produced, for example, in the cytoplasma in case of an infection. And it also happens with proteins that were ingested by the antigen-presenting cell and then transported to the cytosol. This only happens in antigen-presenting cells because they have special mechanisms to make sure that also proteins that were taken up from outside of the cell, we will look at this in the second case, can be fed into this MHC1 loading machinery to make sure we can also make these CD8 T cell responses against these extracellular antigens. So we sum this up. MHC class 1 molecules present antigens which are internal in origin, so they are generated inside the cell, or they are generated from proteins that are generated inside the cell. The peptide degradation happens in the cytosol, and these MHC molecules are presented on all nucleated cells. So all cells that have a nucleus, so there are some red blood cells which do not have a nucleus, they do not have MHC class 1 expression. That's why you can also make blood transfusions very easily, or more easily than organ transfusions. And the function is of these MHC1 molecules to show on the surface of the cell what happens inside the cell. And the effect of the cell presenting these MHC molecules, with the exception of the antigen-presenting cells, is that the cell gets killed because it shows virus peptides. So, if we come to the second case, we have the extracellular pathogens. They are a bit different in how they are fought. So there, the pathogens are taken up actively by the antigen-presenting cell. I said the antigen-presenting cell is like PhD students. They eat everything, including the pathogens also. Then they are inside the cell. They trigger again the activation of the cell. Again, production of, for example, cytokines, co-stimulatory molecules, and peptides derived from the pathogens are presented on MHC molecules, now on MHC class 2 molecules. And this are again all three signals, cytokines, co-stimulatory molecules, and peptides in combination with MHC molecules that are necessary to activate a T cell. Now it's a CD4 positive T helper cell. And once this gets activated, it produces cytokines that then recruit, for example, phagocytes that eat up the pathogen, or B cells that produce antibodies that can then help eliminate the pathogen. Okay, so there's a second class of MHC molecules which are called class 2. They consist of two chains, an alpha chain and a beta chain. There is no beta 2 microglobulin found here. And here both alpha chain and beta chain together form the peptide binding groove that binds the peptide. How do now the peptides end up on MHC class 2 molecules? As I said, these are peptides generated from proteins, usually, that are taken up by the antigen-presenting cell from the outside. So these then end up in endosomes. So endosomes are small bags inside the cell where the material is collected that the cell has taken up from the outside. And these are special endosomes. They are called endolysosomes because the bags fuse with other bags. They are called lysosomes, and they contain a mixture of proteases, so proteins that have specialized in degrading other proteins. And these endolysosomes are a very hostile environment for proteins because of all these proteases, which results in the proteins being degraded, being cut into small pieces, and then we have the peptides. So that's cool. Now we have the peptides. Now we need to load them onto MHC class 2 molecules. The problem is that these MHC class 2 molecules are also produced in the endoplasmatic reticulum. And if we would just produce the MHC molecules in the endoplasmatic reticulum, they would be directly loaded with peptides coming from the MHC loading pathway. So this wouldn't help us because once the peptides from the endolysosome reach the MHC molecules, they would already be loaded because this happens within fractions of a second in the endoplasmatic reticulum. Therefore, we need to block the peptide binding groove of the MHC class 2 molecules in the endoplasmatic reticulum, and this is done by a so-called invariant chain. This is abbreviated with a capital I and a small I. And this is like a chain that hugs the MHC molecule on one side, then it reaches over the peptide binding groove, and it puts just a placeholder peptide in there that just blocks the binding groove. These molecules are then separated from the endoplasmatic reticulum, and they fuse with the endolysosome. And this invariant chain and this blocking peptide are very sensitive to these proteases in the endolysosome. So this gets rapidly degraded, which leads to the peptide, the placeholder peptide, just swimming away because it is not really specific for this molecule. It binds very, very weakly, and it's mostly held in place by the invariant chain. And then we have the peptide binding groove available to load it with peptides, and the peptides are loaded, transported again to the surface of the cell, and can be presented to the CD4 positive T cells. So if we sum also this up, the peptides presented on the MHC class II molecules are generated from proteins that the cell has taken up from its outside. They are generated, the peptides are generated in this endolysosome, these endocytotic vesicles. And the MHC class II molecules are only presented on professional antigen-presenting cells. These are dendritic cells, macrophages, B cells, and some epithelial cells in the thymus and some endothelial cells, which is not so important for us. And the function here is not that the cell gets killed, but the cell that gets activated. And this is the reason because only the antigen-presenting cells present these MHC class II molecules, and they try to educate or find out which T cell is useful and activate this T cell. And therefore, this has the function of activating these cells that then coordinate the immune response against the extracellular pathogens. There are also differences in peptide binding to MHC class I and class II molecules. Here we have a MHC class I molecule, and you can see that the peptides are 8 to 10 amino acids long, and 10 is the maximum already. It's more like 8 or 9. And the peptide binding happens via biochemical interactions between the amino acids of the MHC molecules and the amino acids of the peptide at the end of the peptide. In contrast, the MHC class II molecules can bind peptides that are much longer, 13 to 18 amino acids. And this is the case because this peptide binding groove is not so narrow, but it is more wide. Therefore, the peptides can go outside on both sides, and they can just hang over on both sides. Therefore, they can be a bit longer, and you can see that the peptide binding happens via interaction between the peptide and the MHC class II molecules over the whole length of the peptide sequence. So this is an important difference. So what one can do if one has a lot of time and energy and money, one can take antigen-presenting cells, pulse them with an antigen, then isolate the MHC molecules. As said, if you have a lot of time and energy and money, isolate the MHC molecules and then isolate from these MHC molecules the peptide that are bound, and then you can sequence these peptides. And if you do this, you find peptides that you have the sequence of, and they are very similar. So for example, here you have peptides from the first isolation. You can see they all have the same length, it's the same number of amino acids, and in certain positions you have very similar amino acids. For example, in the 5th position you have a tyrosine, a phenylalanine, and in the last position you have a leucine, and these are the peptides, the amino acids in the peptide that make then the interaction with the MHC molecule. And if you do this for a second isolation, you find peptides that are maybe a bit longer, but you find again on position 2 and the last position you find amino acids that are very similar in their biochemical characteristics, or even the same in all the peptides, because these are the points of contact, if you want to call it like this, with the MHC molecules that need to be similar. Okay, so now I'd like to introduce another term. We talked about MHC molecules all the time, but there's also another term which is called HLA. It is the abbreviation of human leukocyte antigen. And the thing is, MHC is a term that is used by people that work mostly in the mouse system. So a lot of basic research is done on mice. And what people have done in the 1930s and 40s of the last century, they took mice and transplanted skin from one mouse to another. And then they checked if this skin graft is accepted, so it stays on the mouse, or if the immune system of the white mouse attacks the brown skin, and then it gets rejected. And that's how these major histocompatibility complex proteins were discovered. So these are the proteins that are important to decide if a skin graft gets accepted or rejected. Similar results or similar experiments were done in humans. Of course, there you do not just wildly graft skin. That would be not so nice. But what you can do is you can take blood from different humans, and you can just take small drops of blood from different persons and mix them. And then two different things can happen. If the blood is compatible, it will stay liquid. If the blood is not compatible, it will coagulate. So it will form aggregates, and it will turn from a liquid into something that is more less liquid and forming these clumps. And the antigens that are involved in this reaction were called human leukocyte antigens. And once one could study and could clone and isolate and identify these genes, one found out this is the same. Yeah, so the genes that are responsible for transplantation success in mice and for the success of blood transfusions in a way in the end in humans are the same. And therefore people that work in the mouse system will talk about MHC molecules, and people that work in the human system will talk about HLA molecules. So in the following I will just talk about MHC molecules to make it more simple. If we looked at the genetic organization of these MHC genes, they are found on chromosome number 6, and there we have the MHC molecules. And there we have 3 different copies of the alpha chain, which are called HLA A, B, and C. They are not in the correct order. I know this has just to do with their discovery. A was discovered first, and then B, and then C, before knowing where they are located in the genome. And with a certain distance on the same genome, we have a whole set of genes for MHC molecules. You see they consist of a alpha and a beta chain. And the MHC or HLA molecules in the human system are called DP, DQ, and DR. And they always have an alpha and a beta chain coded by an A and a B gene. And for the DR molecule, we have even 2 B genes. One of them is already deactivated in some of us already. So we have these genes here. Then we have accessory proteins. There we have, for example, TAP. We know TAP already. This is one of the pink ones. This is the transporter that transported the peptides into the endoplasmatic reticulum to be loaded onto MHC molecules, MHC class 1 molecules. And then we have some chaperones. Chaperones are molecules that help other molecules to find the correct folding. And chaperone is a term coming from the last century. So the young ladies like Hannah and Alex were not allowed to go out alone and to make forbidden stuff with male persons, and they always had a chaperone with them. This was usually an older woman that made sure that nothing inappropriate happened to them. And here, chaperone in the biochemical sense is a molecule that makes sure that no inappropriate folding happens with these molecules. Okay, so after looking at how they are organized on the chromosome, if you look at the bigger picture, at the whole population of humans, you will find that there are different variants of the genes that form the proteins for the MHC molecules. And you can see here that for the MHC class 2 molecules, there are, as I said, 3 different variants, DP, DQ, and DR. And among these variants, there are at least 350 to 1,200 different variants of the beta chain, and a lot less variability exists for the alpha chain. So these are just different variants in the human population that are able to bind different peptides. And it's even more pronounced for the MHC molecules. So in total, there are more than 6,000 different MHC class 1 molecules. And this allows the whole population of humans to present different peptides from different pathogens more or less efficiently and form immune responses against these pathogens more or less efficiently. So there are always persons in a population that can present, for example, influenza peptides very, very well and do not get sick, and other persons can, for example, present HIV molecules very efficiently and do not get sick from HIV. This is a very, very small percentage that get HIV infected but do not develop AIDS. They are called elite controllers or master controllers, and this is just a way to broaden the protection against different pathogens, not on the individual level, but on the population level. So if we look where this variation comes from, we can sequence a lot of different MHC molecules, and we can check which amino acids differ in these different variants. And you can see that we can divide the MHC class 1 alpha chain into an alpha 1, alpha 2, and alpha 3 domain. And you can see that most of the frequency variability happens in the alpha 1 and 2 domains. And if we look at the beta chain of the MHC class 2 molecule, you can see this is a beta 1 domain on top and a beta 2 domain at the bottom. You can see that the sequence variability is much, much higher for the beta 1 domain. And if we color the respective amino acids in blue that are very variable, you can see that these are exactly the amino acids that form the peptide binding groove. This makes a lot of sense to have sequence variability in the regions that bind to the peptides and to keep the rest of the molecule more or less conserved. So this allows or makes sure that it's able to form the correct three-dimensional structure. Yeah, and as I said, this polymorphism, so the fact that there are many different variants of these molecules that can bind different peptides, makes sure that on a population level we are able to recognize a lot of different peptides from a lot of different pathogens. So two different phenomena come together, and this is usually where a lot of my students have, for some reasons, problem with. The first one is called polymorphism. This means you have more than one variant of a gene. You have a blue variant of a gene and a green variant, and they can be expressed at the same time, codominantly. So then you have green and blue molecules on the surface of your cell. Then you have polygeny, so this means you have more than one gene for a certain function. You can have a red one, a yellow one, and a green one, and they are also expressed at the same time. Therefore you have green, red, and yellow molecules on your cell. And if you combine both mechanisms, polymorphism and polygeny, you can have even more. So you can have for every gene, you can have different variants, and this results that you can have six different variants of one kind of molecule with three different genetical loci, because we have, as you might have known, we have two chromosomes of most chromosomes, one from the father and one from the mother. If we look at how these genes are inherited from one person to the next, one has to keep in mind that they are all on the same chromosome and they are all very close to each other. This means that they get inherited as a package, all of them together. The further genes are away from each other on the chromosome, the higher there is the frequency of recombination, but these genes are so close together on the same chromosome that the frequency of recombination that splits them is very low to basically non-existent. So this one set of genes is called a haplotype, and every one of us has two haplotypes. One is from the biological mother and one is from the biological father. And now, if this theoretical biological mother, biological father have children, their haplotypes get also split among them. So you can see the first child has here both upper haplotypes from mother and father. The second one has the upper haplotype from the mother and the lower haplotype from the father, and so on. So there is a 25% that children from the same biological parents have the same haplotypes. And that's why when we have the need for one of these children to get an organ transplanted, we first look at at the siblings, because there the chances is much higher to find a compatible donor, for example, for a kidney or for a bone marrow transplant. And only if this doesn't work, we will look at more distant relatives like mother, father, uncles, aunts, and if there's no match, then we can look at these registries where thousands or millions of people are registered. And Alex, please, you have a question.
But the mother will never, or the father will never have the exact same MHC molecules, because we have from mother and father, so it's only 50%, right?
Maybe. Usually yes, I would say. Usually yes. But there are also mothers and fathers who realize that they are very closely related afterwards. So this can also happen. And there is only, I said there are 6,000 different MHC class 1 molecules, but there are also billions of people on the planet. So just by chance, you might have the chance that they have maybe out of these, there are 6 MHC class 1 molecules and 6 to 8 MHC class 2 molecules. Maybe 5 of them fitting is already enough for transplantation. But of course, it is much, much more prominent to find this in siblings than in the parents.
Okay, thank you.
Yeah, but crazy things have happened. As brothers and sisters have produced children without knowing. Yeah, this is, yeah. Okay, so what these MHC molecules do is they present peptides in the peptide binding groove to the T cell receptor. And you can see that the T cell receptor not only recognizes the peptide, but it also recognizes structures on the MHC molecules. And this is something we will look at in the next video. And the T cells are trained to recognize peptides only when they are presented on our MHC, our own MHC molecule. So my T cells are trained to recognize peptides on my T cell MHC molecules, whereas the T cells of Alex and Hannah are trained on different MHC molecules. So, as I said before already, we have 3 different types of T cells, and they are also characterized by their expression of either CD4 and CD8. I said this, the T helper cells are CD4 positive, the cytotoxic T cells are CD8 positive, and the regulatory T cells in the end are a subtype of the T helper T cells. So what is now this CD4 and CD8? CD4 and CD8 are co-receptors to the T cell receptor. Here you see them. The CD4 is more like a longer molecule, whereas the CD8 is a more compact globular. So like my fist is the folding molecule, and they are co-expressed on either CD4 or CD8 T cells. That's hence why they are named this way. And they are found next to the T cell receptor, and they do not bind to the peptide binding groove. So they have nothing to do with peptide specificity, but they bind to the sites of either CD4 to MHC class 2 molecules or CD8 to MHC class 1 molecules. And they stabilize the binding of the MHC peptide complex to the T cell receptor. And that's why we have them, to make this binding more stable, more longer lasting, so that the T cell can stick long enough to the target cell or to the antigen presenting cell to become activated or to fulfill its effector function. Alex, yes.
You said that all immune cells produce MHC1, which have a nucleus. And you said also that only antigen presenting cells can activate CD8 positive T cells. So maybe just give a little highlight why only APCs can induce the immune response in CD8 T cells and not other immune cells, because other cells also produce MHC1.
Yes, so the question, if I got it correct, is basically all cells have MHC molecules, but only the antigen presenting cells are able to activate initially activate the T cells. Once the T cell has been activated by the antigen presenting cell, once it is fully authorized to form an immune response, it can start around and kill all the virus infected cells, for example, that express virus proteins on the MHC class 1 molecule. The difference between antigen presenting cells and normal body cells is both present signal 1, MHC plus peptide, but only the antigen presenting cell, and only if it has found a pathogen expresses signal 2 and 3. Only the antigen presenting cell has co-stimulatory molecules and cytokines, and therefore only the antigen presenting cell is able to activate the T cell. This is also something we will look at in the next lecture. Does this answer the question?
This was the question.
Yeah.
Yeah.
Thank you for asking this clarifying question. So, as I said, the T cell receptor not only recognizes the peptides, but also structures on the MHC molecules. And they are trained on our MHC molecules. How this works, we will see in the next lecture. And this leads to the T cell recognition of antigens being so-called MHC restricted. So this means they only recognize peptides if they are in the peptide binding groove of MHC molecules, and only in those MHC molecules that belong to our body. Here you can see 3 different examples. In the first one, we have the correct peptide for our T cell receptor and the correct MHC molecule. The T cell will get activated. The second one in the middle, we have the exact same peptide, but it gets presented on a different MHC class 1 molecule that the T cell was not trained on it, and it does not recognize that. So there's no T cell activation. And in the third example, we have the same, the correct, if you want to call it like this, MHC class 1 molecule, but a non-matching peptide. So this peptide does not fit to the T cell receptor. And also in this case, the T cell does not get activated. So the T cell only gets activated if the peptide matches and the correct MHC is there to present the peptide. So how do the specificities of the lymphocyte receptors now are generated? We talked about it in the beginning already, that there are millions of different T cell receptors. And if we look at the number, it is approximately 1 times 10 to the 8 different T cell receptors. This is around about 100 million of them. And the sum of them is called the so-called lymphocyte repertoire. This is all these 100 million receptors form our repertoire, and this determines what we are able to recognize. So I said it's 100 million roughly. And now I'll ask the 2 of them if they know how many genes are in the human genome. I already mentioned it in the beginning. Alex, do you know?
Yeah, you said 19,500 to 23,000 genes.
Yeah, exactly. So Alex at least is listening to me. Hannah also, of course, I know. And if we put these numbers in our graph, you will see that this is like way, way, way lower than these 100 million. So it cannot be that there is one gene for every T cell receptor. So there must be a different mechanism. And this mechanism was discovered by a Japanese scientist in the 1980s. That was Mr. Tonegawa. And he looked at the T cell receptor. I actually look at B cell receptor, but it's the same. And we are talking about T cells today, therefore I changed it to T cell receptor. He looked at the T cell receptor and looked at the gene, and his initial theory was, okay, there's one gene for every T cell receptor. And then he sequenced the T cell receptor. There was a new technology in the 1980s that you could sequence the genes, so determine the sequence of the genes, how they are made up, of which bases they are made up. And he found that there are not different genes. There's a set of segments in the genome of the T cell, and these are like Lego bricks that can be built together to form the T cell receptors. There are pink ones and green ones and yellow ones, and the cell picks one of these segments and puts them together in different combinations, and that's how these different receptors are made. Crazy. So how does this look? So we have the genomic DNA. So this is the DNA in the chromosome, and there you have if we look at the first chain of the T cell receptor, the alpha chain, It is encoded by the so-called alpha locus. There you have variable and joining segments. It's just how they were called. A big capital V for variable and a capital J for joining. And they're called V alpha for the variable genes of the alpha locus. And there are 70 to 80 V alpha segments. They have like a black leader sequence that is needed for their expression. And then there are 61 J segments. And now the cell, by mechanisms which we will look at in detail in the next slides, picks one of these segments just randomly by chance. So here the third V alpha segment was chosen, and the first J segment would also be the 71th V alpha chain and the 60th J segment, and they are put together. And you can see that the sequence between them, so everything from V alpha 4 to the last one, gets deleted. It gets cut out by recombination events. We will look at the mechanism in the next slides. And this leads to an irreversible loss of genetic information, meaning the cell cannot go back to the initial state and try it again because all the sequence information in between was lost. Then also the other parts that are not needed are cut out. This gets transcribed into an mRNA that gets spliced to get rid of the introns. And then it gets translated into a protein, and this protein then folds and forms the alpha chain of the T-cell receptor. And you can see there is a constant part which is called C alpha that forms like the lower base of the receptor. This is always the same because this is a complex molecule. It somehow needs to fold. If we randomly put everything together, it will never form the correct folding. But the tip of the T-cell receptor, where it recognizes the peptides with, this is then made from these different segments. And this is also true for the other half of the receptor, which is called the beta chain. There we have also V beta and J beta, but also D beta genes. So D stands for diversity, and they are also put together. And therefore, by this set of segments that are put together randomly, we form different T-cell receptors in every cell. And so this is a random event. How many segments do we have? So for the typical T cells, these are alpha beta T cells expressing an alpha beta T cell receptor. We have 70 to 80 alpha V alpha chains and 52 in the beta chain. We have no diversity segments in the alpha chain and 2 in the beta chain, and we have 61 joining segments in the alpha chain and 13 in the beta chain. And there are also additional T cells. They are called gamma delta T cells. They are very special T cells. It's not completely understood what they do and how they do it. You see that there are many less segments, and it's not completely also clear if there are certain segments that participate in actually forming receptors. That's why there is a question mark after the 4. And these gamma delta T cells are important in the gut, for example, to fight pathogens, but they, for example, do not need MHC molecules presenting peptides to get activated. So it's not completely understood. Don't ask me why and how it is. But they seem to recognize mostly lipid antigens. So antigens derived from fatty acids, fatty molecules that are part of certain pathogens. And this is just a second type of T-cell receptor and T-cell type. But the vast majority of T cells are these alpha beta T cells expressing the alpha beta T-cell receptor. So how do we now combine these segments? And this is very complicated and will take us several slides, but I will do my very best to explain it. And we will look at one variable gene segment and one joining gene segment that we want to combine. So this is the V1 is the pink one, and the J1 is the yellow one. And they have so-called RSS sequences, so recombination signal sequences, in front or behind of them. And these sequences are recognized by a molecule that is called recombinase activated gene 1,2. This is abbreviated reg 1, 2, and this has binding sites for these RSS sequences. And what happens? This reg molecule has 2 binding sites. First, it binds the blue RSS belonging to the V segment, and then it binds the green RSS belonging to the J segment. And this results in both segments that were chosen to be combined to be located very close to each other and be fixed to each other in a certain 3-dimensional orientation, so that here the genes can be cut and put together. This bringing 2 RSS stretches together is called forming a synapse. So from this synapse, the reg molecule then cleaves the RSS off, and the RSS stay attached to the reg gene, reg protein, and the segments that we want to combine in this process are separated. And now we have a problem, because we have cut our chromosome into 3 parts. Yeah, the middle part that we want to get rid of, and the first part with the V segment, and the second part with the J segment. And if we now do nothing, everything will flow into different directions and our chromosome will be destroyed. So this would be a catastrophic event for the cell. The cell would die. And this we need to prevent. And this is prevented in the next step. But what we need to keep in mind and what we need to make ourselves aware of is that the DNA strand here is cut to get the RSS out, and its cutting results in different endings of the DNA. For the V and the J segment, we have covalently closed DNA hairpins. So DNA consists of 2 strands, and these two strands in this double helix, and these 2 are joined together at the top, so that the ends cannot flow in 2 different directions. And the other ones are just open, but this is not a problem because they are fixed to the reg anyway. So now we continue. This is basically the same. And now additional molecules bind to the DNA. They are called Ku 78 and 80. 78 and 80 is the molecular weight of these proteins. If you put them on a gel, this is just how big they are or how heavy they are. And these now take on the one side the RSS sequences and phosphorylate them, so they add a phosphate, so they put energy in there. And on the other side, they bind our 2 loose ends so that they cannot flow away. So they are fixed also in position. And then to these Ku molecules, we attract a molecule that is called DNA protein kinase Artemis. This has binding sites for these Ku molecules, and this just results in these 2 loose ends of our chromosome being fixed there in position, and again fixed in positions so closely together that we can put them back together. Yeah, so this just is the way of putting them in the right orientation to each other, that they cannot flow into different directions, and that our chromosome is not destroyed. And what this molecule now does, it opens the hairpins. So you can see that there is a little maybe cut that is made in the DNA sequence, that we have now two separate DNA strands again. And then another molecule is recruited, is being part of this complex, and this is called the terminal desoxyribonucleosyl transferase, TDT. And with the other segments, with the RSS, nothing happens. So let's just put it on the right side that we do not lose it. Then this TDT molecule processes the DNA ends. It puts them together. So it connects the pink V segment with the yellow J segment and ligates the 2 strands again together. And you can see now both ends of the chromosomes are again fixed to each other, but they look different, yeah? Because if you look, there is not just the pink V segment and the yellow J segment. There is also a red, green, and blue sequence in there that was not there before. So there has been some sequence information has been added. On the other side, our RSS sequences are also ligated together. So ligase is a protein that connects DNA molecules. And at the end, we have formed our coding joint and our signal joint. So the signal joint is, if we look at the original germline configuration, is just the RSS sequence together with all the stuff that was between them. This forms like a circular plasmid structure that gets lost during following DNA or cell divisions, because it gets diluted out. And on the other side, we have now the V segment and the J segment very, very close to each other, basically directly next to each other, with a little bit of sequence information put in the middle between them. And this is called the coding joint, because this is then coding for the T-cell receptor, forming the protein of the T-cell receptor. And this is imprecise, yeah? The right side was precise. You can see exactly the blue and the green one with nothing else, nothing more, nothing less. But the coding joint is imprecise because there was information added or even subtracted, as we will see. Okay, just ask the two. They got it. That means somebody else can also get it. It's complicated. Yeah? Yeah, Hannah?
So the somatic rearrangement and the reg-dependent rearrangement only happens for T-cell receptors and B-cell receptors, not for the MHC molecules.
No, the MHC molecules are just taken as they are from their genome. They are also not encoded in the genome as segments. They are fully finalized functioning genes that just get translated into a protein and put on the surface of the cell. Here there is no finalized functioning T-cell receptor in the genome of the cell. It exists only as these segments, and they first have to be put together. And as Hannah just rightfully said, this process of putting it together is called a rearrangement process. And because here a V and a J molecule were put together, it's called VJ rearrangement. If there's also a D segment involved, it's called a VDJ rearrangement. Alex?
I would like to add that this kind of rearrangement we also see in DNA repair. So
yes.
If you're into DNA repair, maybe you know already this mechanism.
Yeah, but they are in the end, what we do is we damage the DNA by cutting it, by trying to cut the middle part off. As I said, it's a big problem to have one chromosome turned into 3 fragments, and then we need to put them together. This also happens, of course, in other processes. It's part of normal life that damage occurs, and therefore these repair processes are also, of course, relevant in other scenarios. But here it is like a damage that gets introduced on purpose in order to be able to recombine these 2 fragments in the end.
In the end, this is a cleverly used mechanism that already exists when you have a DNA repair.
Reuse of resources.
Yeah.
In the end, yeah, exactly. So now I would like to look at this final part in a bit more detail to understand, or for you to understand, how and why this coding joint is imprecise in the end, and how this additional information ends up. Because this is very important. If you would just combine these few hundred segments randomly with each other, we would reach a certain number of T-cell receptors, but we would never reach the 100 million. Therefore, it's not enough sequence. So there must be a process to add even more variability in the T-cell receptor. So if we look again at the stage where the 2 RSS stretches are bound to reg 1 or 2, this looks like this. You have the pink V segment and the yellow J segment, and you have some additional sequence information or part of the sequence information of these genes. And then we form these hairpins at the coding ends, and you see that there's stuff missing on both segments. These are the RSS stretches that are removed, and now we have these covalently closed DNA hairpins. Then, as I said, these hairpins are opened, and when a hairpin is opened, then the sequence just folds over, and we have a longer strand on one side and one that is a little bit shorter on the other side of the DNA molecule. And what we generate here are so-called palindromic P nucleotides. P is just the abbreviation of palindromic, and a palindrome is a short DNA sequence that repeats itself because we have like A binding always to T and G always binding to C. And if you fold this over, if you think about it, this will always result in like a mirror. If you put like a mirror between the first two bases and the second two bases, in this case, the sequence on both sides of the mirror is the same. This is what is called a palindrome. Okay. Then we have the TDT molecule, and this is a crazy molecule that just adds randomly some bases to the end of these palindromic nucleotides. And this is purely by chance, yeah? Puts an A, puts a C, puts a G in there, doesn't care what. It just takes what it finds and puts it in there. So we have some new sequence information added that was never there before, yeah? Only the palindromic information was there before, but the N nucleotides, so these are the non-template nucleotides that were not coded in the DNA before, are added randomly by chance, okay? Then we try to fit these two strands together. This works rather nicely because that's the example I made up, and you can see that from these N nucleotides that were added, there is a stretch of 3 nucleotides, this TA, GC, and AT, that fits together, but the A on the end of the J segment doesn't fit. And this is then removed by endonucleases. So there are proteins that cut off these non-fitting nucleotides. And then we have a partial overlap of these two strands, but it's not a complete double helix is there because there are just holes in there. And this is now filled up by the DNA ligase 4-XR-CC4 molecule. This fills up, checks, okay, there is an A at the end of the V segment. So we put a T, then we have a T, then we put a, and then we have an A, then we put again a T, and so on. So it fills up all the holes and then ligates together both strands at the end. And then we have our segments joined, and you can see we have on one hand you have the V segment, on the other hand you have the J segment. In the middle you have the N nucleotides, the non-template nucleotides that were not coded before. They are new genetic material or new material in these molecules, new sequence information in these molecules. And then we have the palindromic sequences that result from opening the hairpins. So that's why the coding joint is imprecise. And with respect to the V and the J segments, this is sequence information that results in a functioning protein, but the other stuff in between is more or less random, yeah. And in most cases, or in many cases, this results, for example, in premature stop codons, endpoint mutations, or the repair mechanism doesn't always have to work. So this is not guaranteed that this always results in a functioning molecule, functioning protein. Okay, as I said, as this is not always guaranteed to work, the cell has basically 2 chances to do this, because it has one set of segments on one chromosome and another set of segments on the other chromosome. But if it always starts with only one chromosome, trying to put this together in this rearrangement process, and if this is successful, it transmits a signal to the second set that stops the rearrangement of these segments. So it's not completely understood, but it was shown that, for example, the leader sequences and DV alpha sequences are highly methylated afterwards. And this just prevents, for example, the binding of these RAG molecules, and this then prevents that a second variant of a T-cell receptor is put together. And this is called allelic exclusion, that only one set of genes is always recombined. If it doesn't work in the first chromosome, the cell can try again with the second one. But if it doesn't work with the second one, then the cell has, in German we say Pech gehabt. It didn't work, and then the cell usually dies, and then we have to try again with a new cell. That's why we make so many T cells. So there are 100 million T cells made every day in your thymus, and most of them do not form functioning T-cell receptors. So 95% of them are not functioning in the end. And Alex has a question.
You said that we make like 100 million T cells in a day in our thymus.
Thymus.
Yeah. And you said that the most of them is waste. So this is a loss of resources, isn't it?
Yes, and we will also learn in the next lecture that maybe out of the 5% of T cells that make it through the end, a good fraction of them is even autoreactive. So it's like even more dramatic. But the thing is, it's a compromise between losing or wasting a lot of cells and material, but it is like the only way, at least the only way that our body has come up with generating 100 million different receptors from a few hundred gene segments. Yeah, this is just a compromise, and you just take it for granted that most of the stuff you put out is junk. Yeah, this is just a way how to do it. There's no other way. Otherwise, you would have, I don't know, 1,000-fold more genes for T-cell receptors then you have other genes, and you know how many mutations happen over time in all of us, which can lead, for example, to cancer or stuff. And if you have 1,000-fold more genes, you will have 1,000-fold more mutations. There's no way around it. So this is just a compromise in the end.
Okay, thank you.
Okay, so from this allelic exclusion results that one T cell, also one B cell, only expresses one type of T cell receptor. So this is the question that Hannah asked in the beginning. Yeah, there are of course many, several thousand, several 10,000 T cell receptors and B cell receptor on every cell, but they are always the same because of this allelic exclusion. So where does lymphocyte development happen? So as I said, we have this hematopoietic stem cell that develops in the common lymphoid progenitor. This then turns into the T cell progenitor. And here in the thymus, we have pro-T cells that develop into pre-T cell. You see, they have like half of a T cell receptor already because they did this rearrangement on the first chain. Then if they successfully do this, they get some survival signals that they can recover a little bit. Then they try on the second chain of the T cell receptor, and if they have also successfully done this, formed a functional, fully complete T cell receptor, they start to express both CD4 and CD8, and then they get checked if they bind to MHC class 1 molecules better than to MHC, then they are allowed to keep the CD8 expression. If it's the other way around, if they bind better to peptides presented on MHC class 2 molecules, then they're allowed to keep CD4, because in the end, the T cell receptor is a randomly generated structure, and now we just look at what we generated and does it bind better to one or the other type of MHC molecule. And then they leave the thymus as mature T cells, and the same also happens in the bone marrow. So we have a pro-B cell that first puts together the heavy chain of the antibody. It's basically the same from a same progenitor as the T cell receptor, but developed into a different direction. Then we do it for the other half of the B cell receptor, and then the mature B cell is allowed to leave the bone marrow. And just for completeness sake, we have also a pro-NK cell that develops into a mature NK cell, but this happens neither in the thymus nor in the bone marrow, but in the periphery, meaning the rest of our body. So the T-cell receptor complex is consisting of not only the T-cell receptor, but there are many other molecules also involved. So there is CD3 molecules, there are epsilon, delta, gamma chains, and they bind to the outside of the T-cell receptor, but they also have like sequence motifs inside the cell that then transmit the information that a peptide has bound to the T-cell receptor into the nucleus of the cell. And there's also the CD3 zeta complex protein that also is very important for signal transduction. So the T-cell receptor consists of a variable region that is different because it is made from these segments that are randomly combined, and this is needed for antigen recognition. And we have a constant region that is the same for all T-cell receptor that is necessary for folding of the T-cell receptor in terms of the T-cell receptor by itself, and also for signal transduction in the T-cell receptor and all the accessory proteins. So, if now the T-cell receptor recognizes a peptide on a MHC molecule, as I said, the T-cell receptor is not an isolated molecule, but part of a complex with all these accessory proteins. And if the cell also, the antigen-presenting cell, presents co-stimulatory molecules to the T-cell, this results in an activation of the T-cell. And what basically happens is that calcium ions are allowed to come into the cell. They bind to a protein called calcineurin. This then goes into the nucleus of the cell, activates a transcription factor. A transcription factor is a protein that binds to many different spots in our genome and activates or shuts down many genes. And this transcription factor is called NFAT. That is an abbreviation for a nuclear factor of activated T cells. So you know where it is relevant already by name. And this triggers then the activation of many genes. And one of them, one example, the most prominent example, is the interleukin-2 gene. So this gene gets activated, we make an interleukin-2 mRNA, an interleukin-2 protein. This protein gets secreted outside of the cell, and this is a cytokine that binds to its receptor. So interleukin-2 binds to the interleukin-2 receptor, and this triggers a molecule called mTOR, mammalian target of rapamycin, and this is another regulator that drives then the division and the activation of the cell. So this allows all the processes in the cell that the cell needs to divide into daughter cells and to form all these millions of daughter cells that are needed from the T cell to fight the pathogen. The problem now is, as we just discussed also with Alex, is that these T cell receptors, and without spoiling you, it's the same for the B cell receptor, are generated randomly, also generating autoreactive T and B cells. So T and B cells that recognize with their receptor our own cells and tissues, and they would inevitably kill us if we wouldn't have mechanisms to control them. And this is what we are going to talk about in the next lecture. So we're going to talk about tolerance in the next lecture, and we're talking about NK and NKT cells. So if we sum up what we learned today, we looked at T cells, and we learned that they are one of two types of effector cells in the adaptive immune system. So they are also B cells, which we will look at in the next lecture. And we distinguish between three classes of T cells. We have the helper cells that orchestrate the immune response by producing cytokines. We have cytotoxic T cells that directly kill infected cells or cancer cells. And we have the regulatory T cells that are able to shut down immune responses if we no longer lead them, or if we make them, for example, against our own cells and tissues. We learned that the T cell receptors are not coded one by one as final complete molecules by different genes, but they are coded by segments that are randomly combined with each other, also adding some information via the somatic rearrangement. We also learned that the T-cell receptor binds to peptides in combination with MHC molecules, and it recognizes both the peptide and the MHC molecule. And finally, we learned that the T-cell receptor consists of a variable region that is different for every T-cell receptor and is responsible for binding different peptides, and a constant region that then transmits the activating signal. And we also learned that the T-cell receptor is not an isolated molecule, but part of a really complex of different proteins that together transmit the activating signal. With this, we are at the end of the second lecture, and I would ask the two of them again if they have any additional questions.
No, I asked everything already.
Okay, thank you, Alex. Thank you, Hannah. Really appreciate that you joined me for this, again, 3-hour-long recording session. But if you, dear listener, have additional questions, just put them in the comment section of this video, and we will try to get back to you with an answer as fast as we can. Other than that, we thank you for watching. We hoped we could bring a little bit of light in this complex topic of T cells and how they are and what they are and how they get activated. We will continue, as I said, with the next video on tolerance and NK and NKT cells, and we hope to see you all again in the next video with this. Thank you. Stay hungry for knowledge, and see you in the next video. Bye.
Bye.
Because Hannah is more warm, I'm somewhere in the middle, and you look like a ghost.
This is already the warm light, because I can also take this one, and now I am a ghost.
Yeah, because side.
Side.
Side. Side. We have learned that the T-cell. Okay. Das war Alexa. Are we ready? As ready as we can get, probably.
I can take another coffee, but...
Yeah, go take a coffee before we start.
What about the pee-pee break?
Yeah, then we make a pee-pee break, I take coffee, and we start afterwards.
We can manage to this
yeah, that's pee-pee synchronization.
To 30 minutes. Okay, so 4 minutes.
It's not the old guy that wants the pee-pee break. Yeah, I just want to make this clear. But okay.
I just wanted to say, it's not the old guy who wanted the pee-pee break, but it's the old guy who takes the longest.
And he has recorded that.
Yeah.
Now we have a break. Maybe we'll make a pee-pee break.
Pee-pee break again.
Pee-pee break again.
Alex has... Check your prostate. Coffee. Check your prostate.
I don't think that I have that. I just have a high consume of coffee.
Yeah.
Then I can eat my banana. Not on camera. We will look at that in a distant lecture in the past.
Time is not the same concept for me than it is for you. I'm a transcended being as a supervisor. Where do I start again?